Santosh Salunke from Sterling. Thank you for that exciting meeting. Is there similar technology available for IBD surveillance to detect early flat polyps or dysplastic polyps?
You can.
So from my side, I can say that the software is currently developed even for surveillance of patients suffering from inflammatory bowel disease. Of course, this is a very exciting topic, although I feel it's very difficult because we are facing chronically inflamed mucosa, so surveillance should only work when there is really no inflammation, like what we have learned even from the past studies, that we should only do surveillance when everything is non-inflamed. However, you're right, of course, this is something what is also currently under development so far, but not available.
Regarding the collaboration between Cosmo and Medtronic, I can say that, of course, having an hardware solution that is already on the market and is able to host different AI engines, of course, we are working across the entire GI space. Always with the same approach that we wanted to share in the previous talk. So identifying first the bottlenecks for a specific disease and then proceed from that.
Ralph, to you. Yep.
Yeah, so we've seen some amazing technology there, and some very, very impressive images and some very impressive data with 100% and things like this. But at the end of the day, the winners in this technology are always the ones which are practically useful and can be incorporated into daily practice. And the problem comes when you start to make things more complicated and not simplifying the issue for the endoscopists. So I wondered, with Ralph here, who was like the pioneer in confocal endomicroscopy, do you think, confocal microscopy, do you think that the endo-brain is going to go the same way? Is it practical enough to incorporate into general endoscopic practice? And at what point is the technology so simplified that it can be really used by average endoscopists?
It's a very good question. Endomicroscopy is a very powerful tool, but it's very examiner dependent. You see a lot, but the interpretation is difficult. And it also takes some time. If you're trained, you can be quicker, and that's why I'm kind of impressed if machine learning can help you to make the right interpretation. However, and that's then a question for you, with endobrain, the lesion, if you have a 2-centimeter lesion, you really have to spot that area to find maybe the cancerous area. So you might combine first macroscopic view, then going on endocytoscopy, you have staining, so you might add some more time. So you're right, it's from a practical point of view, it might add some time, but it also gives some new information.
Yeah, I think a lot of the technology is really focusing on diminutive polyps. I think that's what the general consensus really has been around the world, because that's what's the biggest burden. So things like endobrain and endocytoscopy for larger lesions, we know that there are limitations related to that, because you have to actually examine all the areas in terms of for a non-expert endoscopist. So for diminutive polyps, touch technique, we've shown, and we're certainly showing with our data, that it is easy to learn, and non-experts have never used it before. Just touch and zoom up. And then press the button, they are able to actually characterize those lesions. But certainly for large lesions, what we would do in our practice is we would use magnification endoscopy and then focus the area of concern with endocytoscopy and then with endobrain.
But the main question would be for a flat lesion, whether there's a submucosal invasion or not. And as I understood you correctly, there's this great video showing that this is invasive cancer just based on the surface analysis. How good is that in terms of accuracy after you did an endoscopic resection or surgery?
So we know even from the magnification endoscopy data that there's good high sensitivity and specificity for flat lesions for magnification endoscopy. The problem arises really is when you have a nodular component, because the surface characteristics of the magnification image don't really reflect what's going on in the submucosa. So decision making regarding treatment is based on the surface characteristics, which really I think is important for the audience to know. Is it's good for flat lesions, but for nodular component, you need to use both magnification assessment, and then you may need to go for a trial dissection with ESD, and then potentially a percentage of those would be more deeper submucosal invasion. You're absolutely right, I think, for flat lesions.
I think I think that's probably you've really touched the sort of heart of the matter, and we're back to asking the same question about training again, and how you guys are high detectors. Your ADRs are in the 60s probably, and I mean yours is 100%. We know, but so how does this you put this in the hands of somebody whose ADR is less than 20%? How is it going to turn them into a fantastic endoscopist? Is it going to save bowel cancer screening for the world? How do we train people who have never diagnosed a polyp with endoscopic diagnosis before? How do we train them still to make a diagnosis without relying on this machine to do the diagnosing for us? Because we've heard that's probably going to get us into trouble. What do you guys think?
Yeah, absolutely. I think that you're absolutely right. And we have all here on the panel developed so many classifications in our scientific life. And but we all learned that this is a problem what you have addressed and also what I've shown in lectures. So it's working for the experts, but it's mostly not working that well for the non-experts. And we also know, although image-enhanced endoscopy is nowadays incorporated in all the endoscopy systems, but still many colleagues are not using it. So our hope indeed is that by using now artificial intelligence and those integrated detection and characterization tools, indeed, we can increase our way to perform endoscopy in the future in order to serve the patients better. We all know about miss rates. We all know about interval cancer. This is endoscopy. This is true life. However, also what is true life is that we are never stopping. We are always trying to develop new things. And in my personal opinion, I strongly believe that now with AI, this might be really now a total game changer.
Yeah.
Quality in colonoscopy will still matter, so you need a clean colon, you need to withdraw slowly, and you really have to expose the mucosa. Yeah. And then all the systems have shown that polyps can be detected. However, you also see that the borders of the lesions are sometimes a little bit more difficult. And also, even if you have the diagnosis, then you have to make the decision whether you do an endoscopic resection right away or you send to surgery and so on. And these will be the learning tasks for the future, because the other things, if you really go on quality colonoscopy with these systems, I think that even somebody who's not so well trained will see with the help of the machine almost all polyps.
Thank you. Audience.
Hi, I'm from a Kentish District General Hospital, and my question really is, does any of these methods have a safety feature saying that, hold on, you haven't investigated behind that fold, let's go back?
There's a Chinese system which can tell you that in gastroscopy, it will automatically go over the whole stomach and tell you that you have seen only 80% and you have to spend more time, and it will make automatically the images of the stomach, which gives you a whole picture. As far as I know, it's not available yet for colonoscopy, but I'm sure that this will happen as well.
Yeah. There are definitely people working on it.
Yeah.
Yes, John.
With these very impressive images and the magnifying colonoscope and all this technology, will AI make the average colonoscopy list shorter or longer for the average colonoscopist?
Longer or shorter? Well, I think that's a really good question. I think often when you evaluate polyps, people that know in our practice, we spend a long time looking at these polyps. 10 minutes or so is not an unusual time for a complex polyp.
And more to take off when you found more.
Sorry, say again?
There'll be more lesions to take off when you found more with the beep.
So I think certainly initially there may be a sense that the withdrawal time, on a quicker withdrawal time, you would achieve a higher adenoma detection rate. But that's going to take us some time to achieve. In the initial phases, I think we'll find that the lists will take longer for the non-expert endoscopists in particular, and then with time we will get quicker. But I totally agree. I think it's a major issue. To sample a certain percentage of mucosa is probably what we need to achieve, which is a combination of, retracting devices, AI, good bowel preparation. We probably need to have a standard of, what is the threshold of mucosa that we need to achieve? Because often it's like looking for a bad blade of grass on a football pitch, doing a colonoscopy.
But I fully agree with your thoughts. I think AI should really do something totally different because, every technology, what we're evaluating is increasing detection and some high characterization as well. And I like your thought because it's time to think outside the box. AI should really improve our practice in endoscopy. And this is something what we need to show in the upcoming years to come. But you're right, it should not start and stop with detection and characterization. There's much more what we can do with AI.
I think there are two main questions. One thing is if the polyps which we identify, are they really relevant or not? And I like your study because it was shown that there were more advanced lesions, when I understood you correctly, to be identified with artificial intelligence. That would be highly
Is interesting. Just not my study. It was an independent study,
Sure.
but also
Your assistant.
regarding I wanted to add that there was no difference in. So I'm not an endoscopist, but I reported that there was no difference observed in the withdrawal time between the AI arm and the other. So basically, this is sort of also unexpected results because one could assume that using AI you could spend more time analyzing the visual cue.
And the second main question is, how many lesions do we miss because we cannot see them, or because we miss them because we have not recognized them? And I think that would be the study, to do tandem studies 2 times artificial intelligence, because the computer will always identify the same amount what the computer will see, and then we know from the naked eye we will miss 20% of polyps if we do 2 colonoscopies back to back. So that will be also interesting to clarify.
Thanks very much, everyone. Thank you. Thanks.